Hello everyone, how are you doing today? This is Abdullah. Today I will be talking about platelets in details. Let us suppose that someone injured his skin. This will result in bleeding, but this bleeding will be stopped after several minutes, 2-3 minutes. Why? Because of platelets and coagulation cascades. Coagulation cascades are many proteins which are reactant and activated each other. These proteins are produced from liver. But platelets are cells which are produced from bone marrow. So we need both bone marrow and liver for making clot or formation of the clot. So in this lecture I will be talking about production, function, structure, adhesion, and aggregation of platelets in details. Let's get started talking about how bone marrow produces platelets. Inside this long bone there is bone marrow, and inside this bone marrow there is stem cells. These stem cells will be converted to megakaryocyte because we need here platelets. But the same stem cell may convert it to another cell to form red blood cell or white blood cell. But in this condition we are talking about how stem cell will produce for us platelets. So this stem cell, which is also called hematocytoblast, will be converted to megakaryoblast, then to megakaryocyte. This megakaryocyte will have several phases, like here phase 1, 2, 3, 4. Let us suppose here phase 1, and this is phase 4. In phase 4, on the surface of megakaryocyte, there is some kind of projection, or similar to villi maybe. This projection will be detached from megakaryocyte to form fragments. These fragments are called platelets. So the platelets are mainly fragments which detached from the surface of megakaryocyte at which phase? Phase 4. So we just knew how platelets are produced. There is some kind of diseases which, for example, aplastic bone marrow or aplastic anemia, when there is a problem with this stem cell. So no platelets, or there's a decrease in the platelets production and red blood cell and white blood cell. So there's some diseases which affect this process. This will bring us to an important information, which is what is the normal count of platelets. The normal count of platelets is between 150,000 to 450,000 per microliter. This is a normal count. Another important information is platelets, sometimes called thrombocytes. So if platelets count will be less than 150,000, this condition called thrombocytopenia. Another important point is that the normal lifespan of platelets is between 7 to 9 days. And after, for example, 7 or 9 days, these platelets will be destroyed inside spleen. So we just talk about production and some information about platelets. Now let us talk about important structure of platelets. Let me draw for you here platelets. This is one platelets. On the surface of platelets there is different receptors which we need to know. The first receptor is glycoprotein 1b. Another receptor is glycoprotein 2b3a. The function of these two receptors, glycoprotein 1b has very important function in adhesion of platelets to the site of injury. Glycoprotein 2b3a has very important function in binding platelets to each other to bring them to the site of injury together, because if there is injury we need so many amount of platelets. So that's why we need platelets to come together, not one by one. So this receptor will bring platelets together. So for example, here there's another platelets. This another platelets will bind to this platelets by glycoprotein 2b3a. What binds them is fibrinogen. So fibrinogen will bind these two platelets together to bring them to the site of injury. Another receptor on the surface of platelets is ADP receptor. Another one is thromboxane A2 receptor. I will talk about this receptors in details when I will be talking about adhesion and aggregation. So these are receptors on the surface of platelets. Another important structure which is inside this platelets is granules. There is two types of granules. There is delta, and there is alpha. Alpha granules will have factor 5, factor 8, and also platelets derived growth factor. Platelets derived growth factor. This one will help in healing of injured endothelium. These two factors will help in coagulation cascades, and also fibrinogen. These are alpha granules. So alpha granules will have several substances. About delta granules will have ADP. This is delta granules. Have ADP, also thromboxane A2, also calcium, and also serotonin. So these are four substances which is existed in delta. This is delta granules. This is alpha granules. This is alpha. This is delta. So here there is some granules inside platelets. So this is the most important structure which we need to know, granules, and also another structures which are the receptors which are existed on the surface of platelets. Now let us talk about platelets adhesion and platelets aggregation. Let us suppose that this endothelium got injured. This will result of production of a factor which is called von Willebrand factor. This von Willebrand factor will be attached to basement membrane in endothelium which is injured. This von Willebrand factor will be produced from healthy endothelium and attached to basement membrane. We will see what this von Willebrand factor will do. So let us suppose that here there is platelets which are going by this injured endothelium. Von Willebrand factor will attach to this platelet. So this platelets will come here to the site of injury. This von Willebrand factor will attach to them by a receptor which is called glycoprotein 1b. So glycoprotein 1b will attach to von Willebrand factor. So here this platelets 1, 2, 3. This 3 platelets became adhered to the endothelium. How? By von Willebrand factor and by this receptor glycoprotein 1b. So glycoprotein 1b is very important receptor for adhesion of platelets. So for adhesion to occur, we need von Willebrand factor, and also we need platelets, and we need also normal receptor which is called glycoprotein 1b. This is adhesion. So we finished adhesion now. Now we will talk about aggregation. These platelets which became at this place, they will be activated. When these platelets become activated, they will produce many substances. They will produce the granules which is inside delta granules, which are ADP, and also thromboxane A2, and also calcium and serotonin. So serotonin will have effect of vasodilation on blood vessels to bring more platelets to the site of injury. Calcium will have a very important factor and role in coagulation cascade. But what we need to know is ADP and thromboxane A2. ADP will go to its receptor and thromboxane will go to its receptor. So they will lead to aggregation of platelets. They will call many platelets to come to the site of injury. So both of ADP and thromboxane A2 will aggregate so many platelets. So let us suppose here there's many platelets. So ADP and thromboxane will bring them to the site of injury. So many platelets came to the site of injury, as we see here. So this will lead to formation of primary hemodynamic plug. So when adhesion and aggregation, both of these two processes occurred, this will lead to formation of primary hemodynamic plug. Why we call this primary hemodynamic plug, both of adhesion, which is the first layer of platelets, and aggregation, because there is secondary hemodynamic plug, which is happened because of coagulation cascades, which are proteins produced from liver. So platelets responsible of formation of primary hemodynamic plug. But here is one point which I need to mention, that as I said, that glycoprotein 2b3a will make platelets together by fibrinogen and bring them to the site of injury. This will accelerate aggregation. This receptor will accelerate aggregation. So we talked about production, function, structure, and also adhesion of platelets and aggregation of platelets. This is very important process, because if we understood this process, we will be able to understand several diseases and also many drugs which work on these receptors. I hope that you got information out of this lecture. See you in other lecture. Bye bye.